spinal cord. The peripheral nervous system is everything outside of that, right? Think about your peripheral nerves, think about your cranial nerves. Okay, the CNS is responsible for maintaining homeostasis. We talked about integrating centers, right? Where signals were received, processed, and then some sort of response was initiated, and that is how we sort of regulate homeostasis. That ability to take signals, figure out what's going on, be it blood pressure, be it pH, be it temperature, make a decision about that stimulus and then send some type of response. We know in the nervous system there are tons of neurons, right? About 100 billion neurons, many, many neurons, and they connect in lots of different ways. Again, that convergence and divergence that we spoke about, this brings about trillions of synaptic communication or connections. Okay, so lots of branching, lots of different connections, different types of connections, and different locations of the body. Now, the nervous system is responsible for basically everything, right? Everything we feel, think, do, emotions, feelings, be it physical or mental, memories, just everything, okay? This list could go on and on. If we have a pleasant emotion, it's perceived with some type of stimulus. If we touch a hot stove, that's perceived using our nervous system. If there's a loud sound, all of that is perceived and transmitted and communicated via the nervous system, okay? So it's a very important system, okay? Yet it's still very poorly understood. It's one of the least understood organ systems in our body. And it's partly because of evolution and partly because we're using the brain to understand the brain. And that's a controversial or difficult concept. And so there's a lot of gray area in behavioral and in neurological science simply because of this, simply because of that relationship. Okay, so there are lots of players here in the nervous system. There are neurons, glial cells. There are many types of glial cells. We'll talk about what they are and what they do. Then there are physical supporting structures. We talked about this in the lab. Meninges, the skull offer some protection there. The blood supply we'll also talk about today. The brain is very sensitive to oxygen and glucose supply, and that happens through the blood. We'll talk about the blood-brain barrier, and then we'll distinguish gray and white matter, okay? Now, neurons are the main functional units of the nervous system. They are both the structural and the functional unit. So the basic function of the nervous system is brought about by a neuron. It's responsible for relaying basically chemical and physical stimuli, all of our stimuli essentially, olfactory, auditory as well. And they are conducted by these electrochemical impulses that we've been speaking about. And so when we look here at this word electrochemical, we're supposed to automatically be connecting that the transmission at the synapse, we talked about that being in two forms, right? That electrical energy of the action potential, but then the chemical bit with the neurotransmitter, right? So this is how we transmit these signals, electrical and chemical at those chemical synapses. All right, so we also have the release of certain chemical regulators, which again we call neurotransmitters. These help to relay those signals, those electrical signals, across from one neuron to the other. And again, this is responsible for perceiving a whole host of stimuli for our learning, right? For our, again, memory formation, glandular control, autonomic function, blood pressure, just everything that you can think about. We're going to look at the autonomic nervous system in detail, and a lot of these functions are not conscious, okay? So sometimes our responses, for example, blood pressure is not a conscious, not sometimes, our blood pressure is never a conscious response, right? So even unconscious responses is what I'm trying to illustrate here, are regulated and transmitted with the nervous system. Now, most neurons can not divide, but they can sort of repair themselves along with the help of some of the glial cells we'll talk about. But it's important to understand that they do not divide, okay? And that is where the limiting capacity in their ability to regenerate comes in. This is why nerves are really hard to regenerate after injuries and such. Now, we can classify a neuron several different ways, structurally based upon the processes that are extending. We can have pseudopolar, unipolar, different shapes of neurons, or functionally depending on what they're doing, whether they're picking up sensory information or whether they're relaying motor information. So we have sensory neurons, motor neurons, and then we have interneurons, which are actually communicating between these two types, right? These are interneurons, which help modulate or regulate sensory and motor communication. Okay, so let's sort of divulge just a little bit more. Now, in terms of the functional classification of neurons, we said sensory neurons perceive sensory information. So when we look at the body and the communication in terms of the nervous system, there's sort of two highways, I like to think of these, in terms of the direction of communication. We're perceiving stimulus from the world, right, in many different ways, auditory, olfactory, visual, and that is one direction of communication. That's from the world to your brain, okay, and that's sensory information. And we typically call this an afferent flow of communication, afferent with an A. Whereas the second sort of highway of communication here is from your brain to the world, and that's a motor communication. So sending out signals for you to move limbs, move your eyes, face, and that sort of thing, it's gonna be in the opposite direction. It's gonna be coming from your brain and going down to that effector tissue, which in this case would be a skeletal muscle. So there are two directions of flow, and then there are two types of neurons that are involved. So again, going from the world, right, your sensory perceptions into your brain and spinal cord are afferent fibers, and these are sensory. Whereas going from your brain and spinal cord with instruction about moving skeletal muscle or about other types of movement is the opposite direction. That's going to be efferent signals. And we'll speak about afferent and efferent a lot when we look at the autonomic nervous system. And then lastly, there are some associating neurons that just help to bridge that connection, okay? They help to integrate the function between sensory and motor. All right, let's look at the glial cells. So the glial cells are the extra non-excitable cells, so they're not excitable the way neurons are. And what type of structure do you think they're lacking if they're not excitable? What structure are they lacking if they're not excitable? So they're lacking an axon hillock, yes, but what channel would they be lacking? That's a good connection. What channel would they be lacking as well? If they are non-excitable, what type of channel would they not have? A voltage-gated sodium channel, right? A voltage-gated sodium channel. So just making that connection here. All right, so these glial cells are of different types, right? We'll talk about the different types of glial cells there are. Astrocytes, ependymal cells, microglia, oligodendrocytes, and Schwann cells. And so we'll touch on their function, their main functions, and where we find them. Now, astrocytes are some supportive cells of the CNS. They're star-shaped, that's where they get their name. They help to bridge the connection between nervous tissue and non-nervous tissue. And so we can see the blood vessels of the brain here being sort of bridged or protected from the neurons are being protected from the capillary or the blood circulation rather. Okay, and so the astrocytes are like a gatekeeper. They're sort of regulating the flow of material from the blood, right? That's the external world. There's viruses, bacteria, other pathogens in our blood. And so astrocytes sort of bridge that gap, bridge that communication, and protect our neurons in that way. They also possibly modulate synaptic activity, right? So they can possibly have this axo-axonic relationship, which can be synaptic facilitation or synaptic modulation as well. Okay, and so they basically communicate through chemical messages here. So they're not influencing the synapse in terms of the membrane potential. They're not giving electrical energy. They are modulating the synapse, and they are communicating or determining how much neurotransmitter is released. Okay, this axo-axonic relationship we talked about. So these are astrocytes, right? Star-shaped. They help to remove neurotransmitters. They help to clean up debris around the area. They are supportive to neurons. Neurons need them in terms of repairing themselves and functioning properly. They maintain normal electrolyte composition of the interstitial fluid in the CNS. And obviously they protect, because of their relationship, because of their gatekeeper sort of location, they protect against any sort of toxins that are in the blood, viruses, pathogens, bacteria, etc. Okay, microglia. Microglia also function with astrocytes, and they protect the brain and nervous tissue from toxic substances, but these are more involved in the immune response of the brain. They have engulfing properties, right? So they can phagocytose, and phagocytose means to basically engulf or to swallow debris or pathogens, bacteria. And so in this way, this is sort of the immune cell of the central nervous system, microglia. They protect the nervous system from oxidative stress. They clean up dead or dying neurons. They engulf bacteria, pathogens, viruses, etc. This is just a bit of a clinical correlation here. We're going to talk about myelin later on, and in terms of understanding some of these diseases and where they fit into the anatomy of the nervous system, when we lose myelin, this results in multiple sclerosis. And this is a slowed or delayed transmission of our electrochemical signals, and that's because we lose myelin. We talked about the function of myelin, we talked about that saltatory conduction, and this is vital for that fast, smooth transmission of these electrical signals. And when we lose that, we end up with some disease process, right? And multiple sclerosis has a lot of different effects. It's immune in its origin, so it's basically the body attacking its own myelin-producing cells. And so it's very difficult to cure because of that immune aspect. Alzheimer's is the loss of cholinergic neurons. So if you lose cholinergic neurons, we get cognitive defects, we get motor defects, and basically sorry, I'm losing my words. The acetylcholine and the cholinergic receptors is one of the most abundant neurotransmitters and receptors that we see. And so to lose this in Alzheimer's, this affects a lot of different structures and functions. Okay, cognitive, movement, as I said earlier. And then finally, Parkinson's, we lose some dopaminergic neurons. Again, so losing these type of neurons inhibits us in this way. Dopaminergic neurons are responsible for movement, okay? And so this is why we have that shuffling gait of persons with Parkinson's. They aren't as easily able to transmit those movement signals because of the loss of some of these dopaminergic neurons. All right, so loss of myelin, loss of cholinergic neurons, which affects cognition and movement, and then loss of dopaminergic neurons. Okay, let's talk about the physical support of the nervous system. This should be a review for us. We talked about this extensively in the lab. The bone of the skull is the most protective aspect, housing the brain, which is very sensitive. That's responsible for or that includes rather the cranium, which is the bony part of the skull, and then the vertebral column, which is the distal bony framework of the spinal cord. Okay, so together, these two structures offer support. We also spoke extensively about these three meninges: dura mater, tougher outer layer, arachnoid mater, more networky, spidery inner layer, and then pia mater, which is the more intimate, closely related to the brain, very gentle or soft layer, indistinguishable from the brain. Okay, and so just to point out those layers here, right, we have the scalp, which is skin and some other connective tissue. We have the skull, which is the bone. Then we have our meningeal layers. Okay, so these are our meningeal layers here: dura, arachnoid, pia being one with the brain. All right, and then within these layers, we have CSF. CSF is cerebrospinal fluid, and this is this fluid that helps to suspend the brain in the cranial cavity. So the brain isn't just sort of sloshed in the cranial cavity, you know, just dropped down. It's actually suspended in this fluid, and so that keeps it protected from those rough walls of the cranial cavity. Very, very important connection here, okay? All right, let's move on. All right, so that's CSF, cerebrospinal fluid, and again, its function is to suspend the brain in the cranial cavity and prevent it from sloshing against the walls. And so it's a clear fluid that helps to bathe the CNS. In terms of its composition, we'll talk about its composition a little bit. It's created from ependymal cells, and ependymal cells line the cranial cavity. We talked about the choroid plexus. This is a concentration of these ependymal cells, and they're secreting this CSF, right? The choroid plexus and the ependymal cells. Again, in lab, we look at the circulation of CSF. It's created from these choroid plexus. It is circulated into the lateral ventricles, then into the third ventricle, then into the fourth ventricle. It's reabsorbed by these arachnoid villi, and then it's recirculated again and again and again. And again, the functions are cushioning the brain and maintaining a stable interstitial fluid environment. Now, when we say maintaining this environment, this is so that it's distinct from plasma. Okay, so we're not just circulating plasma around our neurons, but we pull some of that plasma, we modify some of the electrolyte levels, and we create this distinct environment for the nervous tissue. That's very important, and we'll see some of the ways that CSF differs from plasma. So on this slide. So what we'll see here is that CSF looks a lot like plasma, right? But I don't want you to memorize this. I want you to focus on some sort of the proportions here. There's much less glucose and much less proteins, right? Much less glucose, much less proteins. So when we compare plasma to CSF, plasma is very glucose rich and very protein dense. And so when we look at the function of how the brain uses glucose, how the brain uses glucose, this relationship will become very important. Okay. So again, don't memorize this, but just understand the proportions here. That CSF looks a lot like our plasma, with the exception of the protein levels, right? The plasma is much more protein dense and much more glucose rich.